Greetings to everyone. Welcome back once again to this educational channel on biology. I'm teacher Janet. Today we'll be continue our discussion on nucleic acids. So this topic is from Form 4 Biology KSM syllabus, Chapter 4, Subtopic 4.5. This is the last part of Subtopic 4.5. The title of our video today is Formation of Chromosomes from DNA and Proteins. So this is a short discussion, and after that we will be discussing some important questions on nucleic acids, on DNA and RNA, which will help us to answer questions in the SPM, because some of these questions are also past year questions on DNA and RNA. So stay tuned until the end and watch the whole video. The learning standard for today's lesson is as follows. We must be able to describe the formation of chromosomes from DNA and proteins. So do you know that the chromosomes in our cells are actually made up of DNA and proteins called histones? Right? So here we see a chromosome which is X-shaped. So why is it X-shaped? It's because actually a chromosome starts off as one strand, having a structure of one strand. But when it duplicates before cell division, then it will consist of two strands. Now this is the first strand and this one is the second strand. And the two strands are held together at the centromere, which we'll study in chapter 6 when we talk about cell division. So each strand of chromosome consists of a long strand of DNA, the DNA molecule, such as the one here. Now, so this diagram shows you the beautiful DNA molecule, and you can see that there's a double helix structure. The center part are the base pairs, right? So this DNA molecule in the chromosome is actually very, very long. So it needs to be organized and stored away in a compact manner. Let's find out more about how this DNA molecule is packed and stored away in the form of chromosomes. So before we discuss the formation of chromosomes from DNA and proteins, let us find out some fun facts, which is how long is one DNA molecule? So if we look at one chromosome here, one strand of a chromosome, it contains a long DNA molecule. But how long is this DNA molecule that's coiled up inside this chromosome? So a human chromosome consists of a DNA molecule that is about 5 cm long, or the length of a thumb, the height of a thumb. So just look at your thumb. That is about the length of one long DNA molecule that forms one human chromosome, which is very long because when it's packed up, you can't even see, and because of the way it's packed, you can't even see the chromosome itself, right? So that's how compact the cell has packed that DNA molecule. Now, how about in one cell? If you stretched out all the DNA of 46 chromosomes, so one cell has a nucleus that contains 46 chromosomes. Now, if you stretch out all the DNA of the 46 chromosomes in one cell, it will be 2 meters long, all right, which is about 5 cm times 46 chromosomes. And because some chromosomes are long and some are short, okay, so roughly around 2 meters. So all these chromosomes, if you take out all the DNA and then you stretch out all the DNA and join them all together, they are about 2 meters long, which is about 6.6 feet, and this is the height of a tall person, tall man. So this is very long. This is only for one cell, but our human body contains trillions of cells. So you can imagine how long all the DNA will be if they were all the DNA in all the cells in the human body were to be grouped together and stretched out and joined together, right? By the way, how many genes do you think are contained in all the chromosomes? Now, basically, we have about 20,500 different genes in each cell. But because each chromosome has another copy that's similar to it, because one chromosome comes from father, one from mother. For each type of chromosome, you get one from father and one from mother for each type of chromosome. So we have to times the genes by 2. That means each cell has 41,000 genes. Okay, but in terms of the different types of genes, we have 20,500 genes. This is only an estimate, right? Some sources may state that there are more genes, some may state less. So now we know that each human cell contains 46 chromosomes, which are made up of 46 long strands of DNA. Now this DNA has to be packed up and then stored in the nucleus of the cell. So the nucleus is a very small space in the cell. How does the cell store all this DNA in a systematic and organized way? To understand this concept better, let's take the analogy of a box and some ropes. Let's say you have to store 6 strands of ropes, long ropes, into this box. So there are a few ways of doing it. Now, the lazy man's way is just to throw all the ropes inside the box, and they may become entangled, and then close the lid. So the disadvantage of this method is that it's very disorganized. The ropes may become entangled. Somehow the ropes will take up a lot of space in the box. So a second method that's more organized is to use spools, such as the one shown here, to coil the rope around. So we'll need 6 spools for 6 ropes. So the ropes are coiled around the spools, and then the spools are arranged nicely in an organized way inside the box before being stored away. Right? Now, likewise, it's the same with the DNA in our cells. The cell has a very methodical and organized way of storing this DNA in a compact form, and also so that it doesn't get entangled amongst each other. This is very important, especially in cell division, where the strands of the chromosomes have to separate from each other. Or we say the sister chromatids have to separate from each other. So you find out more in the topic on cell division, about sister chromatids, which are actually the duplicated chromosomes. That's all. So to ensure that the DNA is stored away in a compact but organized way, the DNA is wound around proteins called histones, right? So these are the histones, and they act like the spools for the DNA to be wound around them. So for one long DNA molecule, maybe you need a large amount of histones for the DNA to be coiled around. Now, when the DNA is coiled around the histone, it is called a nucleosome. So nucleosome is just DNA coiled around histones. Then these nucleosomes have to be organized. They are intertwined among each other to form the whole chromosome strand here. All right. Here is a complete diagram showing the formation of chromosomes from DNA and proteins. Let's start with a cell. So in a cell, there is a nucleus. And in the nucleus, there are many chromosomes. So each chromosome is actually made up of a long DNA molecule that is wound around proteins called histones. So let's start with the DNA molecule here. As you can see from here, each DNA molecule consists of 2 polynucleotide chains wound around each other. Here we see the structure of the DNA molecule, which consists of nucleotides. The pink part is the phosphate group, the white part is the sugar, and this part protruding out in the center is the nitrogenous base. Right? So these 2 polynucleotide strands of DNA, they are wound around each other to form the DNA polynucleotide chain. Now, so this DNA molecule is very long, and it needs to be wound around histones to organize it. See, it's wound around this histone, and then since it's so long, the remaining part must be wound around other histones too. So the DNA is wound around histones to form nucleosomes. Each nucleosome consists of part of the DNA molecule wound around histones. Then the nucleosomes are intertwined, so they are arranged and fitted together, intertwined, to form the chromosomal structure here. All right. Now let's come to the notes. So our learning standard is describe the formation of chromosomes from DNA and proteins. 4 marks. Right? You can copy down these notes if you want to. Now, firstly, chromosomes are formed from DNA polynucleotide chains. So the DNA molecule is made up of 2 polynucleotide chains that are wound around proteins called histones. Right? So DNA molecule plus the histone proteins. Now, histones do not contain any genetic information. Next, the DNA molecules combine the histone proteins when they wind around them to form nucleosomes. So nucleosomes are made up of histone proteins and the DNA strands that are wound around them. A part of the DNA molecule that's wound around them. Then these nucleosomes are intertwined to form the chromosomal structure. So what's the benefit of this type of organization or method of packing? The benefit is to save space in the nucleus so that all the DNA can be stored in the nucleus, and also to prevent the DNA, the long strands of DNA, from becoming entangled during cell division. All right. Okay, let's look at the formative practice in the textbook, formative practice 4.5. So I have rephrased some of the questions here, but actually it's the same, right? The first I rephrase it like in the form of exam questions, right? So the first one is name the 2 types of nucleic acids. Answer is deoxyribonucleic acid and ribonucleic acid. Okay, DNA and RNA. Then draw a nucleotide and label its components. So you have to be able to draw the nucleotide, which consists of 3 parts. Normally we use a circle to represent the phosphate group, which is attached to the pentose sugar, right? So for DNA, the pentose sugar is deoxyribose, and for RNA, the pentose sugar is ribose sugar. Then the base is represented by a square, or sometimes it has a shape here that will be complementary to another nitrogenous base, right? So nitrogenous bases include adenine, thymine, guanine, and cytosine, right, for DNA. And for RNA, it's adenine, uracil, guanine, and cytosine. So remember how to draw this nucleotide, which is the basic unit of DNA and also RNA. We also need to know how these nucleotides are joined together to form the DNA molecule and the RNA molecule. All right? Question number 3. Why is a mRNA molecule shorter than a DNA molecule? 3 marks. Now, a DNA molecule contains many genes with many genetic codes or information for the synthesis of many different types of proteins. So let's say this is the DNA molecule here. It contains genes that are arranged in a linear sequence along the strand of DNA. So for example, maybe at the end of this DNA molecule, you have gene A, which is a segment of the DNA molecule. So this gene A has the genetic code for the synthesis of one type of protein, maybe called protein A, whereas gene B has the genetic code or information for the synthesis of another or different type of protein, maybe protein B. And gene C has the genetic code for the production of protein C. So one DNA molecule contains many genes. That's why it's so long, right? But mRNA molecule copies and contains the genetic information of only one gene. For example, gene A on DNA. So the mRNA is formed by copying the code of the genetic information from only one gene from the DNA molecule. So since this one gene forms only a small part of the DNA molecule, for example, gene A, thus the mRNA is much shorter than the DNA. Right, next question. Explain two effects if a cell does not have nucleic acids like DNA or RNA. So this is a negative question, but actually it is asking you about the functions of the nucleic acids like DNA. So when you answer this type of question, you must mention what happens if the cell does not have the nucleic acids like DNA. Don't just mention the function of the nucleic acids. Alright? Now, for example, you will have to start with, if a cell has no nucleic acids like DNA, so it cannot do what? It cannot transfer or pass down the hereditary information that is stored in DNA. And this hereditary information are the genes or the genetic information for the synthesis of various proteins, right? So without DNA, the hereditary information will be missing. So the cell will not be able to pass it down. The organism will not be able to pass down the hereditary information to the next generation. So this hereditary information, which determines the characteristics in living organisms from one generation to the next. Now, secondly, cells will die when they do not have DNA, which actually contains genetic information for synthesis of proteins. Because without without DNA, there's no genetic information for synthesis of proteins, and cells will not be able to produce important proteins for the functioning of the cell and for survival, right? So these are the two effects if there is no DNA. All right, firstly, cannot pass down hereditary information from generation to generation. And secondly, they will die because they cannot produce the proteins needed for the functioning of the cell. Look at another question that is similar to the one found in your textbook. That is in Summative Practice 4 on page 82 of the textbook. The question is phrased like this. Question 2b. Complete the diagram below to form a whole DNA molecule. 3 marks. Now, this question is similar to the SPM question that has been asked before in the old syllabus, where the student is required to draw the DNA molecule. But part of the molecule has been drawn for you, so you just have to draw the other part. So we know that a DNA molecule consists of 2 strands of polynucleotides, right? So one strand is shown here, and in this strand of polynucleotide, 2 nucleotides are shown. The nucleotides are the basic units of the DNA molecule. So here we have one nucleotide, and this one is the second one here. All right, so in the first one, we can see the phosphate group here, then the pentose sugar, and one nitrogenous base. In this case, it's cytosine, C for cytosine. And for the second nucleotide, it has the base adenine. So now you're supposed to draw the other part of the DNA molecule, which is the other polynucleotide strand. You have to draw it here on the right-hand side, right? So you can pause the video for a while and then try to draw the diagram yourself. And we'll discuss in a short while. Right, let's see how to draw the diagram. First of all, you must draw the polynucleotide chain or strand facing downwards. Now here we see the strand. You look at this pointed part of the pentose sugar is pointing upwards, right? Like an arrow pointing upwards. So for the second one, we say that it is anti-parallel to the first strand, meaning that it's parallel, but it is in the opposite direction, facing the opposite direction. So it should be like this. All right. Now you see the pentose sugar, the pointed end is now facing downwards. This one is facing upwards. This one must face downwards. So if you draw the pentose sugar similar to the first chain here, first strand here is wrong. It should be pointing downwards, not upwards. So one strand points upwards and the other strand points downwards. for the pentose sugar. Next, there must be so if the strand is drawn correctly, first of all, with the pentose sugar pointing downwards like this, then you get 1 mark. Secondly, to get the second mark, you have to make sure that you write the correct complementary base. For example, complementary base of cytosine is guanine, so write the G here. And complementary base of adenine in the DNA molecule is thymine. Okay, now that is the second mark. For the third mark, the examiner will look at the shape. So here, the shapes must be complementary. For example, this shape is rounded, protruding out. So this one should protrude inwards, and it should be a rounded shape too. Similarly for this, these two shapes should be complementary or matching. That is just to show that these are complementary bases. All right, these two bases are complementary bases, and these two bases are complementary bases. So there's 1 more mark for the shape of the bases. Okay, so altogether 3 marks. Finally, the last question in summative practice 4, page 82. So I've rephrased the question, and it's a similar question. So again, this has been asked in the SPM question before, right? Now, state 2 differences between A, the structures of a DNA molecule and a polypeptide molecule, 2 marks. So each mark is for 1 difference, or 1 difference is equivalent to 1 mark. And B, the structures of DNA and RNA, of the molecules of DNA and RNA, another 2 marks. So let's start with the 2 differences between the structures of a DNA molecule and a polypeptide molecule. Now, DNA molecule, can you imagine how it looks like? Yes, it has a double helix shape like that, and it's made up of 2 strands of polynucleotides that are twisted around each other. And how about a polypeptide molecule? Can you imagine how it looks like? All right, so polypeptide molecule is made up of a long chain of amino acids. So now let's try to explain the differences. Now, what's the first difference? A DNA molecule is made up of 2 polynucleotide chains. Okay, now these 2 orange chains that consist of the basic units called nucleotides. Whereas a polypeptide molecule here consists of a single long chain of amino acids. That means we are comparing the basic units of DNA and the polypeptide chain. For DNA, the basic units are the nucleotides, whereas for the polypeptide molecule, the basic units are amino acids. In fact, this answer has 2 differences already, but actually you have to separate out if you want it to be considered as 2 differences. The first difference is that the basic unit of DNA molecule are the nucleotides, and then whereas for the polypeptide molecule, the basic units are amino acids. Now, the second difference is that the DNA molecule is made up of 2 strands of polynucleotides, but the polypeptide molecule only consists of 1 single strand of amino acids. So we can combine together to write like this. Then if you want another difference, so this one, the first one, 3A1 is considered 1 difference. And 3A2, a DNA molecule has a double helix spiral shaped structure. So double helix means it's a spiral shaped structure consisting of 2 strands of polynucleotides. But a polypeptide chain does not have a helix or spiral shaped structure. It is not spiral in shape, although the polypeptide chain can be folded to form a 3-dimensional, the 3-dimensional shape of a protein molecule. Okay, but it is not a spiral shape or does not have a helix shape. So these are the 2 differences between the DNA molecule and the polypeptide molecule. Let's go on to the second question. 3b. What are 2 differences between the structures of DNA and RNA? Now, this has been discussed in the previous video, so here we are just writing it out again. So there are many differences between RNA and DNA in terms of the structure, which we have to know. Now, the first one is DNA consists of 2 polynucleotide chains, right, as shown here, that are twisted around each other. How about RNA? Can you imagine how a RNA molecule looks like, or a mRNA molecule looks like? Yes, it looks something like this. It's single-stranded. So DNA consists of 2 polynucleotide chains, but RNA consists of only 1 polynucleotide chain. Next, a DNA molecule is definitely much longer than an RNA molecule, because DNA molecule contains more genes. RNA only copies 1 gene, the genetic information of 1 gene from DNA. Now, thirdly, DNA molecule has the base called thymine, the nitrogenous base called thymine, whereas an RNA molecule does not have thymine in its molecule, but it has another base called uracil. Alright, and then lastly, DNA molecule contains the sugar called deoxyribose. That's why the whole molecule is called deoxyribonucleic acid. Whereas a RNA molecule has a different sugar. It has the ribose sugar. That's why it's called ribonucleic acid. So easy to remember this difference. If you remember the name of DNA and the name of RNA, you'll be able to state this difference as one of the differences between DNA and RNA. Now, is it possible to see the DNA in a cell? Well, if the DNA is all clumped together, since it's a very long molecule, a macromolecule, if all the DNA in the cells were to clump together, you'll be able to see them. All right, like what we do here. So there is an experiment that can be done even for school science projects. And this has been done in my school, where we obtain fruits from different types of fruits. We get the different types of fruits. And for example, for strawberry, we can extract the DNA first by mashing up the strawberry and by adding certain chemicals. Now, all this must be done under supervision by a teacher, because it involves some chemicals. So by adding some chemicals and carrying out some processes, finally, when you leave the mashed strawberries or fruits inside the petri dish or beakers, the DNA will start to emerge as white masses like this. Alright, so all these are the DNA molecules that have clumped together. The white stuff are the DNA from the fruit cells. For example, here we can see the DNA from the strawberry cells, okay, emerging slowly, though not complete yet. And this is from orange, and this is from kiwi fruit. Okay, so the main purpose of mashing them up is so that all the DNA can come out from the cells, and then through various processes, the DNA will emerge and be seen in the fruit juices that are produced, in the pulp that is produced. Right? So this is a very interesting science project, and sometimes it's done even at the Form 6 level. So that's all for this lesson, and goodbye for now.